running late from Hello.
Hi, Hua.
Hi, Jeff. Sorry, I'm running late from the.
Okay, can everyone see Anita's screen?
Yes, we can. I can see it. Yeah.
Okay.
Hi. Do you want to start us off maybe for a second, Jeff, or would you like me to just start talking?
Well, yeah. I mean, I think we have everyone now. So I mean, the call was sort of bring Anita into sort of what Elsevier has been doing. And, we can also provide, some updates on what bioCADDIE is doing. So this is, sort of a, I guess, not a getting to know you meeting, since I think most people know you, but sort of, understanding, sort of what each of the projects is doing.
Yeah, that's
really
just
great. If I can make just a quick background comment. Anita showed this to me last week at the NDS meeting in San Diego, and I brokered the attachment to Phil so that he could see it. He went back and forth with Anita on this, and we thought it a good idea to have the bioCADDIE core development team take a look at what Elsevier is doing here, so that they're aware of what these other activities might be, how they might contribute to what bioCADDIE is trying to do with regard to the prototype, and whether or not there are possibilities for leveraging each other's activities in the pursuit of this first prototype data discovery index for the NIH Commons.
Right. Exactly. Thanks, Ron. And just to be absolutely, completely clear, and in the interest of full disclosure, as I think most of you know, when the original call for the Data Discovery Index went out, we collaborated with Marianne Martone and a number of other groups, Melissa Handel, and also the Carnegie Mellon Group, to write a proposal for this. And that was the beginning for our interest in research data search, and it became a question that we were just very intrigued by. How do you make a search engine for research data? And so what I'll show you, then of course the bioCADDIE proposal got funded, but we were still interested in this topic, and we have been working since then to look at what we think a research data search engine might look like. And I think I've just been joined, which is wonderful, because both of them said they wouldn't be available, but by Jez and Tony, is that correct? And they both might be on mute. I think they're both with their children in other places.
Yeah, I'm on, Anita. So, yeah, hi, everyone. This is Tony.
Thank you. Brilliant. Thank you. And I see Jez, too, and I don't know any other Jez's, so it's probably going to be Jez Alder, who's our user-centered design guy, and Tony is our architect. So they're both in the UK, where it's something like 7 o'clock, so they're out in public, so they're on mute. But just want you to know that they're on board and they're on the line. So what I'm showing you is really what we have done since we last saw everyone at the Data Discovery Index workshop, which I believe was that a year ago at NIH?
The initial workshop was in August 2013.
Oh, it was
Is that what you're talking about?
Yeah. That might be. Well, after it had been clear who had received the awards and such.
Oh, okay. The award went out in September 2014.
That's what I thought. Yeah, I figured it was about a year ago, and I think I met a lot of you then. But we didn't quite have the prototypes that we now have. So I just wanted to show this to you because I think we are still very interested in working with the NIH. We're interested in working with you. And we, as Ron said, we're very interested to see, I think our efforts might be very complementary. So we're very interested to see to what degree some of the work that we do and that you do might still be done together and maybe look at opportunities for future pilots or something. So without further ado, well, there is further ado if I could give a quick intro to all the things that Elsevier is doing in terms of research data management, but I don't think I will. So if you're interested in context, maybe I can do that later if we have enough time at the end. But perhaps I should just jump into a demo. And I think this URL has also been sent around. Is that all right with everyone?
Sure.
Okay. So my demo is coming from my colleague Helena Kuzijn. She's a neuroscientist who is working inside Elsevier on research data management. And I said, what are you interested in? Whoa, there's some very loud noises. And they're very good mute.
Yeah, we'll try to mute ourselves.
Thanks. All right. So,
hi, Anita, Caroline.
Oh, hi, Caroline. Hi.
Hi. Do we have a UK access number? Because I can't get in via my desktop, and I've just had to dial the US code, and I've dialed the usual UK WebEx, and I can't get in on it.
Right. So this is the DDI access. Do you guys have a UK access number by any chance?
I don't know if that's set up for this or not.
I have no idea. Kathy, can you answer that?
Oh, yeah, sorry, with our account, we only have the call-in via the VoIP, I think, or so.
Yeah.
You could potentially use Skype to call in.
Sorry, Caroline. Otherwise,
okay.
We've got a bunch of people on the call, so I think we'll be good. And
okay,
we'll keep you posted.
Okay, I'll catch up with you later.
Thank you so much.
You. Bye.
Thanks so much. That's great. Sorry, where were we? So this is the homepage of the data search project, and I've just entered a name that might be relevant in life sciences, but I could also enter something in. And let's see, what was a good query again? Yeah, Jez, I think you have some very good queries. Like there's mantle data. Let's see. So just to show you the types of sources that we have. And the types of queries that we're doing. So for the project, what we've done is we've indexed Dryad, the Harvard Dataverse, biodegradable magnesium, there you go. Well, I'll show this one first, now I'm here. Dryad, the Harvard Dataverse, PetTB, which is an earth science database. We have looked at the open access collection off of PubMed Central. We've indexed a number of journals on Science Direct, and a material science database at NIST. And so what we've done is we've indexed these sources and are rendering them in a single interface. And when I say indexed, I'll show you what it looks like. Let's see if we, so just had a good query right there. Let me go back to the, all right, mantle calcium variation. Let's do that one. So really we're making this look as much like Google as we possibly can. So if you look at mantle calcium variation, this is a typical query that you would find in the earth sciences. Then you can see that we're rendering the results from these different data sources, and we're looking at different data types as well. So for instance, here's an image from Science Direct, and what we've created are these so-called data previews. So this is an image. The images and tables inside of Science Direct we're treating as data objects. Then we're showing a preview of that image in this case. If we look at the full data set, we see that this is actually a journal article that has a number of tables and images related to them. So we can step back and forth and we see here this information changing. So this is tabular data from a particular paper. In the description, we see the caption of that particular image set. Here are the data authors, of course, and then here's the figure description in this case. So we can step back and forth between the items in this collection, where in this case a collection is a journal article.
Okay, so
on
Anita,
the other hand,
within Science Direct, the search you're doing is across the article, and then you're returning all the content from that article?
So in this case, the search we're doing is across the tables and the figures in Science Direct and in PubMed Central for those two databases. for Dryad and Dataverse, which are more generic, right, and we're, as Jen says, we're indexing the table contents. So we're looking at the table itself, the caption to the table, and also the way that the table is being referenced inside the article. So the sentences that pertain to that table.
Okay.
Does that make sense?
Yes, yes.
Okay, great. For Dryad and Dataverse, oops, no, I'm not sure what I did there. Oh, I went back. Sorry. For Dryad and Dataverse, we're looking at the records inside the data repository. So if I go to Dryad for a second, they have tabular data inside Dryad. I can show it here. And this item within Dryad is only a data table. And so in any case, we can go to the data sets, which in this case will give us that particular item inside Dryad. If we look at PubMed Central, similar to Science Direct, we've indexed the tables and the figures as well as the references to the tables and the figures, taking the XML of the open access component of PubMed Central. So again, here's an article that has a number of items, which in this case are all images. Ooh, I'll click that one away very quickly. In the case of ThermalML, which is a database that's created by NIST, we have indexed their full database into the search engine interface. So here we have a property set that consists of tabular data, and these are created by NIST. And similarly by PetDB, in PetDB, PetDB is a geochemical database. And we're looking here at what the PetDB representation of this data is. We're also mapping it, so we're showing it on a map. PetDB classifies this. These are tables that have been indexed to certain articles, but we're also indexing this by sample type. So in PetDB, there are different samples, and then we're looking at the data for one particular sample. So I'll pause for questions for a moment there. I don't know if there are questions at this point. Okay, so I'll continue briefly. We're also indexing all these content sources by data type. So there are documents, file sets, images. Raw data can contain, for instance, zip files that we can't recognize. There are slides. Statistical data can be data sets like SPSS or R. There's a great amount of tabular data, so all the tables that are there, and then a number of videos. So these types, if we can identify them, we'll have them here so that you can select the data type. And these are mostly, the videos are mostly in Science Direct, I believe. Oh, and sorry, we're also identifying the supplementary data. So some of these video files can be supplementary data to a record. And lastly, we are looking by data, by the date that the data set was created. So there's apparently a very old one here, and that's kind of interesting. Most of them are much newer, but so you can identify at which dates was the data set created and filter by that. And then in all the cases, as mentioned before, you can either look at the preview or you can go to the data set directly. And so this, once the article renders, this will bring you to the right figure in that particular article. Let's see which one was that, figure 5. So once it's rendered, it should go to figure 5. It hasn't rendered yet. There we go. So that'll jump straight to figure, boom, 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 boom. Here it is, figure 5 is the corresponding figure of that, and it should jump to that once you go straight to that data set. So that's basically what we've done, and what we're doing at the moment is we're very interested in adding more sources. We're also very interested in expanding the types of filters that we have. So we'd be very interested to talk with you to see in your user community what types of filters do people use and what types of topics would they like to filter on. And we're also interested in developing interfaces that are really useful for specific communities. So the one that we're focusing on right now, as you can tell by the content sources, are earth science and material science. But we're very interested to know in life sciences what are the types of interfaces that people are interested in, and also how this all compares to what you're doing. So I don't know if there are any questions, or perhaps it would be great, Jeff, to see the demo that you're working on, and maybe we can then discuss possible overlaps or synergies or something like that.
So
any questions, maybe, to start there?
Anita, this is Waard from UT Health. I'm just wondering, like, is all the connection to those objects about tables and the figures, is it open, or is it only for Elsevier science director that you can actually run? Is this kind of something open to? I'm asking this, is just thinking if later on something like this, we try to add some function like this, is it possible for us to access this kind of table figure data, or it's kind of closed?
That's a good question, and actually Phil Bourne asked the very same question. So at this moment, for the Science Direct tables and figures that are not open access, you can't access the tables and figures. There's a lot of discussion ongoing whether or not supplementary data will be made open. We do have links to databases, so those are certainly available. And of course, all the open access content you can access. And Elsevier has a bunch of open access journals as well. I'm not, but yes, you can preview them here. Yes, exactly.
How about the PMC articles? And do you know if they have kind of API let you link to tables, figures within the PMC central, PubMed central repositories?
Yes.
Oh, okay.
All other databases are accessible, just the Elsevier ones.
So, Anita, this is Ron. I think we mentioned this when we talked last week, but germane to the point that Phil was asking and Hua as well, as bioCADDIE becomes integrated as an indexing search engine within the Commons is based on open access. Mm-hmm. So that is a central principle to the FAIR concept. So that, I think, needs to be borne in mind.
Yes, absolutely. Well, this would function in largely the same way. So you can certainly preview the data tables and images through the data search demo, absolutely. And all of the rest of the data search demo is accessing open content. The one question is, if you click on that, if you go to the data set, can you then look at the table within the context of the paper? That's the only question at this point for this whole demo, where the access at this moment isn't open for all the records. Okay. But all the rest is completely accessible, yes.
Yeah. What is your guys' plan about this? Are you going to make it open access to everyone who can search through this? If you are building something like this, the easy way for us is actually we say in bioCADDIE we have a link. When we click that link, actually goes to your web API to get additional those figure data information. That's just one possibility I'm thinking about.
I'm not sure I completely understand what you're proposing.
Oh, what I'm proposing is actually first I ask a question whether this data search eventually it will be available for public users. Like
so
anyone can
we're using public data sets, and we have an interface to that.
Okay. That's cool.
It's not that there's no real data in here. We just have an index with a user interface.
Okay. Yeah. So what I propose is say for example in bioCADDIE, if we search something and it's related to a figure or table data, we actually can connect from bioCADDIE to your web search interface for users to access the figure and the table data. kind of like if later you have some kind of API, we can actually link from calling that API from bioCADDIE website. That's something I'm thinking about.
Right. Yes. So it would be really good, perhaps at this point, if you have a demo at the moment.
Yeah, we can do a demo.
It would be great. Maybe we can understand better what it is that you're working on, and then we can maybe see how that might.
Instead of building our own kind of figure data search, maybe we can actually link that to your website. Yeah.
Okay. So it'd be great to see what you have at the moment, and I gave up the control of the data
Jeff,
search.
are you doing the demo, or we should do the demo? Maybe.
I mean, since you're doing the UI development, you should probably demo your UI. And I mean, Anita, while they're getting that set up, I mean, the development, just to give you a little background, is sort of split in two parts. So my group here at UCSD is doing the sort of the back-end pipeline for the indexing, and that's working with the output from Suzanne Sansone's metadata working group, you know, to sort of define sort of the metadata attributes for the data that's being indexed. And so that's what we're working on here. And then Hua's group is working on the UI component, you know, that sits on top of the index data.
And what content sources are you indexing?
So right now it's things like PDB, Geo, BioProject, you know, dbGaP, things like that. But really, it's you know any data repository out there, you know, that we can gain access to.
So, are you focusing on NIH repositories or also on others?
Others as well. I mean, PDB is not NIH, so I mean,
oh, sorry, I didn't know that.
This is Ron again. The bioCADDIE has, you know, if you go back to the white paper, they were given certain marching orders essentially by the NIH. So among the things that they have to be working on is including indexing of data from the centers of excellence, which are developing their own data, and a couple of other of the NIH projects within BD2K. So they're starting with readily indexable data that's pretty structured, and we'll move on to some of these other sources as well as it continues to develop the prototype.
Right, and so the data is not hosted by bioCADDIE. It's data that you index that is hosted elsewhere. Is that correct?
Correct.
That's right. We're not hosting physical data. We're actually hosting kind of metadata description of those data and the link to the original data set. Right.
And so you have a metadata standard, and you then add that metadata to the data sources. Is that correct? Or they add it themselves and you share a standard?
Right now, we are doing, basically Jeff's team is doing it. Jeff can describe that.
Yeah, I mean, the data sources provide the, I mean, so for example, PDB provides, you know, their XML version of the information. Geo uses, you know, minimal, you know, which is a standard, you know, for the microarray data sets. And so we're pulling data from their sort of standardized data descriptors.
Right, and then you host the metadata.
Yeah, we have a cached version of their metadata. Yeah.
Yeah, just parenthetically, with regard to standards, there is a separate effort ongoing, funded just recently by BD2K, to set up a standards coordinating center to pull together standards that can be then accessed by bioCADDIE and other BD2K projects.
Okay, so there's a data standards sort of center as you explained. Okay, that's great to know about also.
Jeff, how do I get access to share our screen?
Oh, do I need to do something here? Hold on.
You can assign Anu the presentation roles, I think.
Hey, Jeff, if you just click on the little ball and pass it to Anu, that should.
There you go. Now we are presenter, yes.
So.
Can you see our screen right now?
Can you see the screen?
Not yet.
Do something maybe? Share my screen.
Okay.
Right.
Oh, yeah.
Okay, now do you see it?
Oops. Yep. It took over my screen.
Great. Got it.
Anita, can you see it?
Yes. Thank you. Yes.
Yes. Oh, you can just. No? Okay. Go ahead. Then Brilliant can. Yeah, you can just type something. Yeah. So basically, this is the first page of the bioCADDIE website right now. We actually have 7 repositories with indexed with 4 different data types and number of data sets. And the second in the middle shows those 7 data repositories we are having, like Geo, PDB, dbGAP, other things I couldn't read very Array Express, things like it. Basically, a couple of them are actually the gene expression data sets. And the basic, we can just go to the top, type of search and something, whatever. Okay, so we have this is a kind of little bit new feature trying to show some facets right over here when you're typing. And yeah, I can just click. So on the left, you can see the different repositories. And like similar to what you showed, you can check one and check one to filter to which database. And yeah, when you click one of the databases, it's actually showing just the data from dbGAP right now. And we also have more facets within the one particular data type. For example, over here, yeah, it has this restrict about data access, right, consent types, things like that. So for each individual data repository, we actually have more kind of data type specific facets. On the left, you can actually narrow down with the search. And in the middle, basically, it's the individual data set you found. And if you click one of the data sets, it will actually show you more details about the data itself. And we have this PDB mapped to the metadata? No. Which one was mapped to the metadata?
Go back to PDB.
So, sorry.
Oh, go ahead. Go ahead.
No. If you could go back to the record where you just were with the PDB. I just had a quick question, which is probably quite trivial, but yeah, if you could, yeah, there you go. So.
Oh, yeah.
Oh, yes.
Yeah.
So these are the metadata fields that you add?
Yes, that's what
correct?
I'm trying to say. This is the metadata, actually generic metadata description coming from the working group led by Susanna. Jeff's team actually mapped the individual data source to this kind of generic metadata schema. And we actually,
I see.
Yeah, showing here. I think it's actually the data metadata schema is publicly available.
Yeah.
Yeah, to go through the working group 3 site.
Yeah, if you want, we can send you a link, or you can find it online as well. I'm pretty sure.
That would be great. That would be very interesting to see.
Yeah. So basically, this is some kind of individual data set. And on the right, we are trying to build something like a similar data set on the right column, like a similar data sets and something more about like a literature related to this data set. Although right now it's just like a prototype because it's really taking time to find the accurate similar data set and related articles. So basically this is the individual data set. Let's go back to the list. So additional function, we integrate several, like a pilot project like this one, RC Delph, which is a kind of a more kind of visual analytical way to view the data set. After you type a keyword, maybe return 200 related data set. And this one actually will do some like a word cloud clustering, actually showing you some more kind of visual analytical for the data, the whole data set returned. Doing some analysis among the, for example, the entire 200 returned data set. This is
so these
pilot project.
Sorry, for my understanding, so these are the data sets that you have selected through the interface where you just were, and then you...
Yeah, so this is, for example, if we type lung cancer search in the bioCADDIE and it returns, say, 300 data sets, and this pilot project actually will run some analysis on the 300 returned data sets, some word clouding, some clustering, and trying to do a little bit of summarization on those entire 300 relevant data sets. That's just like a one pilot project coming from one of the group.
Okay, but these are, it looks like these are rendering papers, is that correct? It looks like these are papers at the bottom. Or?
It's not paper, it's more about description text about the data set.
Oh, I see.
Yeah.
Yep.
They initially developed this for papers, but we sort of took this and implement for the data sets now.
Okay, right.
Yeah.
Right.
So there's a couple of other pilot projects also integrated, like one is about searching GWAS data, things like that. Basically, you can, instead of just searching the name, you can search in the GWAS sample size, platform, things like that. This is also another pilot project was integrated to this bioCADDIE project.
So here, these are publications, or
those are publications actually, yeah.
And there you index the whole publication, the full text of the publication.
Index abstracts, and only something related to GWAS study, I think, for that pilot project. Because I'm actually leading that pilot project, so each pilot project is for a specific data type, and it's kind of small scale. We just implement this to bioCADDIE to demonstrate all different uses of those pilot projects. It's probably still a little bit far away actually to apply all those pilot projects to all different data types, because that needs more work.
Right. That's totally clear. And so.
Yeah, pretty much that's it, right? Jeff, did we miss anything?
Well, I mean, I think the issue of what's being indexed. I mean, for most data repositories that we have looked at so far, I mean, they all have standard metadata, sort of the standard Dublin core subset, with the name of the data set, a description or abstract, authors and things like that. And then there's additional information. So PDB has additional information on material entities such as chemicals and the like that one can add to the record. A geo has potentially other information related to genes, and so it's this sort of adding some of the additional context to the data sets based on what specific information they have. But it all starts around sort of the core data set description. And that's all sort of laid out in that Working Group 3 document.
Right. And so is your goal to add more content sources provided that they have the correct metadata? Is that a good order to state, or to add more content sources and then to find the correct metadata no matter where you can find it?
I mean, it's to bring in more content sources. I mean, most content sources have, that we looked at have the baseline metadata, sort of this, Dublin core minimal subset. But there's also a later working group where we'll be interacting with different repositories. There's also another work group, what is required to come into bioCADDIE. So there might be, some discussion with the repositories as, what type of metadata they should provide. But right now we're looking to bring in some more content so that, we have, the ability to, again, to look at a few different communities in terms of, how they want to, again, search through the data. And that's the work that Hua and his group, are going to be, directly involved with.
Yeah, I think on the metadata, like ingestion part, actually, Jeff's team, we're also talking about actually two different ways. One is Jeff's team actually actively working on more data repositories. And the other attempt we just started is Jeff is working with George, trying to see if a data repository want to convert data to our metadata representation, how can they do it by themselves? So Jeff is also provide the documentation to external sites. So George's team will be one of the first attempt to, as like external data repository, how they can convert data by themselves.
Right. This is George Alter.
Yeah, George Alter.
George Alter at the ICPSR.
Yeah.
Yes. Okay. Okay. That's great to know. So, well, thanks very much, and it's very interesting to see and great to see how much progress you made. So, I'm wondering. I mean, I can imagine two directions perhaps of work, and one would be that we would look at your metadata schema and see if some of the content that we have could be expressed in this schema, or what it would take to do that, so that you could access it. I mean, I'd certainly be interested in exploring that. I can't immediately upfront say we can definitely do that, but certainly for some of the links to our papers and the links between the data sets and the papers, I'm thinking that might be possible. But it would be good to take a look at it. If there's anywhere you can point us to, then we might be able to see how we can make the metadata available in a way that might work within your platform. I mean, the other question is, so that's perhaps a clear, straightforward way forward. If we can look at that, if you could send us a link to the metadata type and tell us what it takes to be included.
Yeah, Anu will send you the link.
That'd be great. And maybe we can explore that and get back to you. And of course, you're interested in life science data, and it looks mostly, is it true that you're mostly interested in genes or other types of life science data interesting as well?
Yeah, I think we're open. We're not limited to gene. Any kind of biomedical data set is within our scope. And yeah, I think we are very impressed by what you have, and it looks like really two systems very complementary. And if we can link to each other, that will be actually very nice.
Yeah, that would be very interesting. So my other question was whether, so as you see, we focus really on these data previews, and we're really looking inside the data sets if there's some way to preview it, and then also allow access to people who come from very different directions. So they could be coming from agriculture, or they can be coming from some other field, but might be interested in seeing some of the data sets here. So I was wondering if there's any way that you're exposing your, if you have any forms of APIs to allow access to the data that bioCADDIE is. Sorry, there's lots of really loud noises. but if there's any other way that we might be able to index some of the data that you're representing.
So I mean, the data, there will be and there is right now sort of an Elasticsearch endpoint where you'd be able to sort of get the index data from.
Okay.
If you're talking about getting access to, you know, to the index information.
Yeah, I mean, I think that might be interesting. We could do something that we don't immediately put on our our full prototype, but we could explore to see what it would look like if you want, if you're interested in that.
Yeah, I mean, the other question, I mean, gets to sort of, you know, is there going to be, you know, what level of, you know, interchange of information, you know, among sort of these, you know, would be useful.
Yeah. So one thing I was wondering, I saw that there was that there are still some pilot project calls out there, and one thing that we could do is to say, well, we could sort of jointly see if we can form a sort of a pre-pilot proposal group, because I understand that you're on a very tight schedule and you have a lot of deliverables that you have to make. So I want to be very mindful of that and not take any time out of your development trajectory. But would it make sense perhaps to explore in a little group whether we might identify a joint pilot where we can say in greater detail, here's your interface, here's the type of work that you do, here's our interface, here's what we do, what might be some ways that we could work together. Do you think that might be useful given what we've shown each other so far?
Yeah, no, I think it would be useful. I mean, the question is how that you know, because I could definitely see, you know, ways that we can interact in terms of, you know, exchanging information between the indices.
Yeah, I think we are very open to any collaboration. I just want to make sure I understand the pilot project you're talking about. Which pilot project you are talking about?
Oh, I don't know. I thought there was a call for pilots, but maybe I'm totally wrong.
Oh,
I was wondering whether it might make sense for us. I understand.
Anita, they have periodic calls for pilots, and they just recently had one. There'll be another one, I think, later this year, Jeff, from Hua, if I'm not mistaken. Every 6 months, I think, was the planning for pilots.
Yeah, we just had 3 projects, right? Harvest.
Right, right.
Yeah.
Right. So the topics, I think, can be fluid. So, Anita, what you could do is, based on this discussion, come forward with a proposal for a pilot that might be useful to tie together what bioCADDIE is doing within BD2K and how it might connect to what is going on in the private sector or academia.
Yeah, I think that makes a lot of sense. So maybe we just missed the mark, but then we can make the next one. You're saying they're every 6 months?
That's
well,
right.
yeah, I mean, and Ron, I mean, there's also, I mean, I don't know if you know, if Bill or someone else is interested, or, you know, this is also, you know, potentially some supplement, or, but again, that's, I mean, there have been many pilot projects funded,
that
right?
tends to be fluid.
Yeah.
It'll depend on the budget. It'll depend on how much is in that sort of pool for supplements and what the competing demands are. That is, there need to be compelling topics. And so if bioCADDIE came forward with a compelling topic that's not being covered by through the current work scope and budget, then it's possible to take that to BD2K and say, okay, we have this compelling interest with a potential idea that could solve this problem or that problem, and we need a supplement in order to do it.
Yeah. That would be a supplement.
Are you looking for funding to fund a pilot project, Anita?
We're not. No, we're interested in collaborating with you.
Right. So I don't know if this needs to be a call for your typical call for supplement or your call for pilot. Those are funded projects, Ron.
Yes, that's true.
I was more looking on the bioCADDIE team side, because I know that they have a tight development trajectory, and what I was trying to do was avoiding to giving the impression that we would ask for lots of extra work to be done on top of what you have already agreed to do. And that's really why I raised the topic. But we are not directly looking, for reimbursement on our end. We're just interested to work with you and explore what it looks like to make a really good search engine in life sciences.
Right. So what you're asking for is there an opportunity to be able to work with our core development team to accomplish
Exactly. Totally right. And work and sit and meet and all of that. You know, I don't have any ideas about definite outcomes, but I think already in a brief discussion we can see that there are many different ways in which we could work together. And I just want to make sure that that is not seen to be something that isn't funded under the current bioCADDIE project. That was my main concern for raising this, but you're totally right.
Well, bioCADDIE does have as part of its charge outreach to the community, and as what the community may have to offer can leverage and synergize with what bioCADDIE is trying to accomplish.
Yeah, I totally agree. I think, like I said, we are very open to any kind of collaboration, because and what you are doing is very relevant to what we are doing. I totally don't see any reason why we shouldn't work on this together. And we have weekly CDT core development team meetings. So if we can identify something specific we're going to benefit both sides, then we can work on that. What do you think, Jeff?
Yeah, I think the first piece would be to, potentially how to sort of create a sort of interchange between indexes, because I'm not sure exactly what the format is. So the metadata scheme that we're using is is documented in that work report, and we're working to to get the indexes to conform to that, and then in terms of back-end technology, we're using Elasticsearch, so all the index data will be available. So I'm not sure, Anita, on your end, if you have a specific metadata model that you're using, and also.
So we're not. We're using Solar as the search engine.
Okay. Yeah. So.
But Tony, who was on the call, but I believe he's in the swimming pool, so you have a lot of background noise. He certainly started by looking at Elasticsearch, and in fact looked at indexing some of our table content in that way. But I think we do have engineers on our end who would be able to look at your schema and see what types of mappings would be useful to do in either direction. I think it would be very useful for us to see how is your content structured and what aspects of our content would really mesh well with that, and then we can have a next conversation where we say in a bit more detail, and maybe we can even play around with some code or something to say what it would look like.
Yeah, this is Tony. We've not used any particular overarching standard. We're obviously trying to accommodate the different sources we're encountering, primarily for the purposes of the limited field-based searching that we're doing right now, and more heavily on the presentation side, so that we have the right fields to display the information. But yeah, we're only talking about a handful of fields at the moment, so it's not too much on our side. Obviously, the main challenge we have is adapting to everybody else's standards that they're using to provide a unifying model for us.
Yeah, and I think, for instance, it would be great to access the metadata that you have that might help us with some of the filtering steps that we do, and it would be great to use your taxonomies for that, for instance. Just to take a look at how is that content enriched, and might that be something that we could also render in our interface? That would be tremendously useful, perhaps. So perhaps the best, we're reaching the top of the hour, but what might be really useful is if we could possibly take a look at your metadata standards or any endpoints that you have. And at the same time, I think that you got a copy of our data search tool. So feel free to look around and see if there's any components that particularly interest you. And then maybe we can plan a follow-up meeting once we've taken a bit more of a look at both of our backends. And if we don't need to have a separate final project, that's great. If you're just able to work with us within the scope of bioCADDIE, of course, that's great. Would that be good? So we'll exchange. So yeah, we'll send the link to our demo.
The metadata, the pointers to the metadata working group documents and all that, and then also we can give you, we're working on bringing some data and re-architecting some of the way we're generating the indexes, but then we can send out that information as well.
Yeah, any information you have would be great. And as I said, I'll also talk for a bit because we have a number of this is separate from the data search interface, but we have a number of data journals that might be very interested, that really focus around a data set with a little bit of write-up around it. And so this is slightly different tangent, but they might be very interested to make sure that they comply with the metadata standards that you're providing. And be able to be indexed by bioCADDIE as well. That might be a different link. So starting there would in any case be great to see how you index these things.
Yeah. I think we can list a few areas we can collaborate on, and starting from there, decide how to move this forward.
Yes, I think that's good. Maybe we can have a couple of emails back and forth, and then plan our next meeting after that. Mm-hmm. I think there's plenty on both sides to be gained by collaborating, so that would be great.
Okay.
All right. Okay, thanks. Any other points from people on the call?
I don't know what. I wanted to know, Anita or Jeff or Wa, if you want to invite Elsevier in for one of the next few core dev meetings, maybe in a month or something.
Which meeting?
One of your core dev meetings.
The weekly meeting?
Yeah.
I think maybe after we email each other, identify the areas, maybe either it could be allocate like 20 minutes from the weekly meeting to discuss it, or we find another ad hoc time particularly for this collaboration. I think either way is fine. Yeah.
Okay. All right. Thanks, Anita.
Thank you so much, and thanks so much, Stephanie, for getting us together.
Hi.
And Ron. Thank you very much.
Thank you, Anita.
All right. Thanks very much. Bye-bye. We'll be in touch. Thanks.
Yes. Bye.
Bye. Yes. Bye.